When the molecule is ready to be exported out of the ER, it gets packaged into a transport vesicle. This vesicle is made of membranes from the ER itself. The transport vesicle travels to another organelle known as the Golgi Apparatus. The main function of this organelle is to finish, sort, and ship cell products. It works like the postal department of the cell. Structurally, the Golgi consists of stacked, flattened sacs, like a stack of pita bread. The Golgi receives transport vesicles that bud off from the ER and contain proteins. It takes the substances contained in these vesicles and modifies them chemically in order to mark them and sort them into different batches depending on their destination. The finished products are then packaged into new transport vesicles, which will move to lysosomes, or be inserted into the plasma membrane, or dumped out of the cell if the protein is a secretory protein. Here is a video on protein trafficking through the Golgi. Some proteins from the Golgi apparatus are transported to the lysosomes. As the name suggests, a lysosome is an organelle that breaks down other substances. Lys means to cause destruction. They consist of hydrolytic enzymes enclosed within a membrane. Hydrolytic enzymes break polymers into monomers through hydrolysis. Lysosomes may fuse with food-containing organelles called vacuoles, and then the enzymes digest the food, releasing nutrients into the cell. Protists do this. Damaged or unneeded proteins may become enclosed within a membranous vesicle, which then fuses with a lysosome. The organic molecules from the breakdown process are recycled and reused by the cell. Chloroplasts. These organelles convert solar energy to chemical energy through photosynthesis. Chloroplasts are partitioned into three major compartments by internal membranes: thylakoids, stroma, and the intermembrane space. Mitochondria are referred to as the powerhouses of the cell. They convert chemical energy, glucose, into a more usable and regenerative form of chemical energy, ATP, via cellular respiration. Mitochondria have their own DNA that resemble the DNA of bacteria, not the DNA found in the nucleus. Plant and animal cells have many common features, shown in red ovals. Cell walls and chloroplasts are unique to plants. While both plant and animal cells have a vacuole, it is much larger in a plant cell. Vacuoles are membranous sacs, and they come in different shapes and sizes and have a variety of functions. Central vacuoles store water, strengthening and making the cell rigid. Food vacuoles provide a place for food to break down. Contractile vacuoles expel water from the cell. The cell wall is an outer layer in addition to the plasma membrane and is only found in fungi, algae, and plant cells. The composition of the cell wall varies among species and even between cells in the same individual. All cell walls have carbohydrate fibers embedded in a stiff matrix of proteins and other carbohydrates. Plant cell walls are made of the polysaccharide cellulose. Fungal cell walls are made of the polysaccharide chitin. Animal cells also have a cytoskeleton made of tubes of protein, giving them internal structure and shape. The dynamic cytoskeleton allows animal cells to change shape and crawl on surfaces, something plant cells don't do. Click on this image to see the cell crawl. Can you see the cytoskeleton moving? The cytoskeleton is stained yellow/green and red in both of these images.